Good morning to everyone, and thank you for your presentation. Our project aims to develop new CFTR modulators for the treatment of basic defects. In particular, 508del mutation can be corrected by the use of small molecules. Correctors, on the basis of their mechanism of action, can be divided in at least three main families. Only compounds belonging to different families can produce a synergistic effect when combined together, and this is highly desired to have an high rescue of CFTR. For example, Kaftrio, which is the standard of care of CF patients, is a combination of two correctors of type 1 and 3 respectively, and one potentiator. And although several advances have been obtained for the treatment of CF patients with Kaftrio, the discovery of new compounds acting as new modulators for new combinatorial treatment could be beneficial for CF patients. Supported by the Italian Cystic Fibrosis Foundation, we started from first discovery of a candidate having a tricyclic structure named PP07, which showed the corrector activity in the YFP-SA on CFB41- cells expressing the 508del mutation. This compound, interestingly, showed a synergistic effect when used in combination with VX-809, indicating a possible complementary mechanism of action. From there, we started with some chemical modification with a medicinal chemistry approach, and we transformed this compound, obtained some analogs, among which compound PP8, which showed improved potency and efficacy in the rescue of CFTR. Its activity was confirmed in primary epithelial bronchial cells from homozygous patients for a 508del mutation, and as expressed in the panel CND, this compound confirmed the high synergism and potency in the rescue of CFTR, especially in combination with VX-809. From there, we started a wide campaign of chemical synthesis and structural modification with a medicinal chemistry approach to establish the structure-activity relationship of our class of correctors and understand which were the main features required to obtain corrector activity, following the workflow showed in the slide. We obtained several active compounds, which are shown in this summary, in this figure, which summarizes the YFP-SA results for many compounds. But we selected compound PP28 as lead candidate for its promising activity. In particular, this compound short-circuit current recordings on human bronchial epithelial cells for F508del mutation for homozygous patients showed high potency comparable to VX-809 and showed a strong synergism when used in combination with it. From the traces, it can be seen that this compound is very well tolerated and that a strong rescue of CFTR with an increase of 8-fold of the ion current was detected. From the dose-response relationship shown in this figure, it can be observed that PP28 showed comparable activity to VX-809 at the maximal efficacy concentration indicated by the green arrow, and when used in combination with it, its EC50 values decreased to nanomolar level. At protein level, by immunofluorescence analysis, we observed that the treatment with VX-809 produced an increase of expression of CFTR, mainly at the perinuclear region, with respect to control cells, where the CFTR red is intracellular, whereas the treatment with PP28 produced a strong increase of the CFTR expression, mainly localized at the plasma membrane level, indicated by the arrows, and this effect is strongly increased with the combination of the two correctors. To have an insight on the mechanism of action, we also studied the combination of our compound PP28 with other correctors. In particular, we used the class 2 corrector 3151, and we compared with the combination with VX-809. As can be observed from the figure, both compounds were able to produce a synergistic effect, producing an increase of the quenching rate. Whereas when PP28 was combined with a class 3 corrector for 172, only VX829 was able to produce a synergistic effect, but the effect was not observed for compound PP28. Thus, we compared the activity in combination with VX445, and as can be observed in the lower panel, the combination of the three correctors for VX445, 4172, and PP28 all produced a synergism in combination with VX829. Sorry, slide. With the all produced synergism in combination with VX829, but the effect was not observed when they were combined together, meaning that all these correctors belong to the same class, acting as class 3 correctors. During the years, we have collected and identified several active derivatives, which are summarized in this figure of the YFPSA. Each dot represents one compound. Many active compounds were identified, even with efficacy higher than the combination of the two vertex correctors. In particular, we recently identified an improved compound named PP209, with respect to PP28, which in the dose-response relationship show nanomolar activity as a single agent, and in combination with VX829, its EC50 value decreased to 170 nanomolar. The activity of this compound was confirmed on human bronchial epithelial cells from homozygous patients for F508 del mutation, and as can be observed by the traces, the compound is very potent, already at 1 macromolar as single agent, and in combination with the VX829, it produces a strong rescue of CFTR, increasingly the ion current, indicating a really high rescue of mutant CFTR. During our chemical exploration, we also approached the scaffold doping strategy to obtain compound with a more flexible structure, which could be useful also for the modulation of medchem properties. We generated a second family of correctors sharing the same mechanism of action, named by us SH compounds, which were progressively improved through different rounds of synthesis, reaching to a good candidate named SH157A, which showed activity at a low macromolar level. Although less potent than PP compounds, there is much space in this class of compounds to obtain improved derivatives. The activity was also confirmed in primary cells of CF patients, and as can be observed, the most important, the most precious feature of our compounds is conserved, that is to say, the ability of synergizing with VX829, indicating that the scaffold doping strategy has not affected this feature. Considering the wide number of possible structural modification of our compounds, we used the support of a rational approach by molecular modeling studies. Considering that cryo-EM microscopy demonstrated that VX445 lies at the surface of the CFTR, establishing crucial contacts with the elastomotive and the helix 11, but also with the helixes 2 and 10, in the attempt to understand the binding mode of PP compound in the binding site of VX445, we used a docking approach by Schrödinger Maestro. We aimed to understand the position and orientation within the VX445 binding site. Many poses were generated and evaluated by scoring functions, and even though we found lower values with respect to VX445, it was observed that PP compounds established important chemical interactions with the helix 11 and the elastomotive, so the same binding site of VX445. Moreover, we compared the activity of PP28 with PP209, and in the docking analysis, we observed by visual inspection that this compound is able to generate an additional hydrogen bonding with an hydroxy residue in the tyrosine residue of the helix 10, being probably responsible for the improvement of activity. To better understand the interactions between our compounds with CFTR, we schematically divided then the core structure of PP and SH compound in three main regions shown in the slide. From the data collected so far, we know that the presence of a sulfonyl group in the region C is essential for the interaction with the lasso motif in analogy to VX445, but also that the presence of some specific chemical groups in the region A are important to establish interactions and to have corrector activity. So, inspired by the improvement of activity of PP209, we generated a family of fluoroderivatives shown in the slide. This is a summary figure that summarizes the result of the YFP assay on CFB41- cells. Each dot represents one compound in combination with VX829 at a concentration of 1 micromolar and normalized for the activity of VX829 alone. The dotted line represents the activity of VX829, and the blue dashed line is the efficacy of PP28 in combination with VX829. We divided our compounds, about 60 analogs, in different groups according to the substitution in the benzyl moiety, in a specific benzyl moiety, having fluorine derivatives or in combination with other groups. And as you can see, we obtained many active compounds, many of them also improving the efficacy compared to the combination of PP28 with VX829. In particular, two derivatives are very promising in terms of potency, and these bear an additional methoxy group in combination with a fluorine atom. And also, we recently found two derivatives indicated in blue, which come from the manipulation of the region B, less explored so far because we thought that it was not really important for corrector activity. And this is a new milestone because this gives the space for the improvement of the activity, and this region could be useful to establish further interaction to CFTR, but also for the introduction of groups which could modulate the medchem properties. In this figure, we also report some candidates which are very promising for the activity. These figures summarize the effect of selected compounds on human bronchial epithelial cells homozygous for alpha-8 del mutation. In particular, those in the green boxes represent the best candidates, which are very potent as single agent, but in combination with VX829, they produce a strong increase of the ion current, indicating a strong rescue, functional rescue. In particular, compound 2T6, as you can see from the figure, from the traces, is very potent alone, and in combination with VX829, it produced a 5-fold increase of the ion current. To better understand the interaction of our compounds with the CFTR, we use also a ligand-based approach by Ligand Scout to understand the main features needed for the corrector activity. And in particular, we selected, on the basis of the additivity index, 30 of the most active derivatives, and we processed this compound. We obtained 9 main features for the pharmacophore model of PP compound, meaning that these are crucial points to obtain activity. The pharmacophore model was then compared to that of VX445, and from the alignment of the two pharmacophore models, we got an important information that our compound needs side chain elongation to improve the activity, and this, in our hypothesis, could correspond to an increase of the distance between the regions A and C, which could, from one way, improve the interactions with CFTR, but at the same time improve the the alignment with the binding site to CFTR, which could generate an increase of potency and function. Together with evaluation of potency and efficacy, we also evaluated at the Istituto Italiano di Tecnologia the medchem properties of selected candidates. 5 compounds were selected on the basis of the structural diversity. I report here the stability on human microsomes and mouse microsomes. All compounds showed very high human microsomal stability, and interestingly, compound SH157A showed also very high stability in mouse. And thus we indicated this compound for further evaluation for pharmacokinetic studies, which will be assayed at the Facility of Translational Pharmacology on mouse. These power compounds will be dosed at 3 and 10 milligrams per kilo and by oral and intravenous administration. Different samples will be collected at different time points, and then the concentration in the tissues will be measured. At the moment, we are waiting for the Ministry of Health approval to start the study. In conclusion, we are still ongoing with our multiparametric optimization of our compounds, caring about potency, safety, but also selectivity of our compounds, and we aim to improve all these features. At the moment, we have so far synthesized 450 derivatives between the 2 classes of correctors, and we aim to select one candidate for preclinical studies. I would like to thank the 2 research groups involved in this study, many of which are in the audience. I would like to thank Dr. Tiziano Bandiera for the support, and as a SAM member, but also as a friend. And I would also thank the delegations adopting the project. These are the last delegations, but I thank also all the delegations adopting the project during these years, and all of you for the kind attention.